I published the latest version of my newsletter, Longevity Trends, a few days ago. And this time it's about skin aging and what one can do to have a better skin. Not only cosmetically, but also related to aging processes. Like when you get older, you have an increase in the risk for skin cancers. You also have an increased or decreased, I should say, wound healing. And obviously, those are things that have mortality and morbidity consequences. So it's not just about looking good; it's also about protecting your skin from the harmful effects of aging. Yeah, you can read it. It's a little bit long this time, but it's I hope an interesting read. And next month, I think I will do it on the exposome. So all the factors that are influencing our aging process. So all the environmental factors and exposures we are exposed to. And this is an upcoming field where in the past we identified exposures, like, we looked, oh, okay, maybe air pollution is related to aging, and then we went to look for air pollution and so on. But in recent years, the idea is to take a population and really track everything they get exposed to as much as you can, and then see what comes out of it. So, a little bit like big data, like the omics data we collect, but then on exposures. So in silico medicine has raised over $100 million. So that's good news. A bit bad news is that the antibody against Alzheimer's disease from Eli Lilly, Kisunla, has been removed by, or has been voted down by the EU for approval, and they cite safety concerns, and that is the amyloid-related imaging abnormalities. And basically what it means is that in the brain, you get too much inflammation as a consequence of these antibodies. So, I mean, it's the idea of these antibodies is they...
no.
Oh, the idea of these antibodies is that they bind to the amyloid plaques, and when antibodies bind to anything, they activate your immune system, and the idea is that the immune system destroys these amyloid plaques. But of course, what can happen when you do that is that the immune system gets too active, and that causes damage, and that is what you see here. So a certain number of people who are treated with this drug have this as a side effect, that the immune system reacts too extreme to the antibody, and that the damage caused by the immune system is worse and can even lead to death compared to the benefit gained from removing the amyloid plaques. So it's that balance that is off. A bit better news then is that there was a paralyzed man who was able to stand up again after receiving reprogrammed stem cells, so iPS stem cells. Now, this was done only in 4 people. One person was able to stand, 2 people had some improvements, but this is a very small sample size, and we have to be cautious because, of course, some of these patients may also just naturally get better over time, right, rather than as a result of the treatment. So it's too early to draw conclusive results, but it's a hopeful idea. And then here in Dublin, I will go to that, so you can meet me there. So here they look at a class of drugs for sarcopenia. And so these are drugs that influence the neurons, the motor neurons. And they show that when you give these drugs to mice, you see an improvement here in, for example, wait, here it is. So yeah, a 22 to 24% enhancement of the rotarod. So that's a rotating rod where the mice have to stay up, and you see how long the mice can just stay on that rod. And also here a 17% increase in the max weight that the mice can pull, and a 12% increase in grip strength of the old mice. So quite a nice improvement. Here, this paper identifies that in old muscle cells, branched chain amino acids, So those are like isoleucine and so on. The ones that sometimes people who go to the gym take as a supplement, these ones are not broken down sufficiently in old people. So in young people, actually taking BCAAs will enhance your muscle strength because they are necessary to build the proteins that your muscles are built of. But in old people, these cells actually accumulate the amino acids, and they just accumulate, not as proteins, but just as amino acids. And they are not broken down or being used to make proteins from. And this accumulation actually leads to an activation of mTOR because these amino acids are very strong activators of mTOR. And then actually this activated mTOR causes problems such as reduction in autophagy, which is necessary for the muscle strength to for the muscle cells to remain young. So what you basically have here is a defect in old muscle cells that then leads to chronic activation of mTOR and the negative effects of that. Yeah, here another paper on muscles. And here they show that the overexpression of BCL-XL, that is an anti-apoptotic protein, is actually higher in centenarians compared to younger people. And if you overexpress it in mice, you have a better muscle function. So here they show that SIRT5 is also good at improving the muscle function in older animals. Here they look at medications and they're linked to muscle bioenergetics. And so they look at metformin and statins, and they found that these drugs decrease oxidative phosphorylation. So that's a process by which you make ATP in the mitochondria, and the electron transport in the mitochondria and the maximum ATP production are being decreased in men, but actually not in women. So there is a sex effect here happening. So this is a clinical trial in humans with metformin for in old people for muscle function. So these people are relatively old because the average age was 80.4 years. So pretty old people who had very slow walking speed. And these people were given 500 milligrams of metformin 3 times a day for 4 months. And basically there was no improvement in these people. But of course, it is already extremely old people. So the question is, would you expect any benefit? For example, we have seen in worms as well that when you treat old worms with metformin, you don't get the benefit you get from treating young worms, for example. So it's possible that these people were already too old to receive any benefit. There were adverse effects in 100% of the people receiving metformin, but also in 92% of people receiving placebo. So that's yeah, yes, maybe also not interesting in that sense. So here, and the problem is, yes, okay, now I can see my title. So here they give senolytics to older people who are at risk for Alzheimer's disease. So they give a combination of dasatinib and quercetin, 2 days every 2 weeks for 12 weeks in total. And they showed some improvements in cognitive function. The effects were relatively small, but they were there. It's also a relatively small study. So, it's a pilot study. So further studies with more people will be necessary to confirm if indeed the senolytics are having an improvement effect on Alzheimer's. So here is another Alzheimer trial, a phase 2A. So before the big phase 3 trials in which you will demonstrate clinical benefits. So again, we have to be careful in that it's not meant to show improvement. It's more to show safety. With antiretroviral drugs, so drugs that prevent retroviruses from being active. And in our genome, there are these retrotransposons, also known as jumping genes. So they are probably old viruses that over hundreds of thousands of years ago to millions of years got inserted into our genome and have been ever since there. And when you get older, these retrotransposons can become active. They are normally repressed by epigenetic mechanisms, but when you get old, these epigenetic mechanisms start to fail, then these retrotransposons can become active, and then they can cause genetic damage because they start moving around in your genome. And so they insert in other places in your genome and they can disrupt genes and things like that. And so they gave this inhibitor of these retroviruses to people. And they had 12 people in this trial over 24 weeks. Again, it's mainly safety and tolerability, less if it actually works. And so they found a favorable safety profile. There were stable cognitive measures. Of course, of over 24 weeks, you will not expect big changes in cognition. And yeah, there were no further problems. So this it suggests that there was reduction in neuroinflammation. So again, not a primary outcome of cognition, but a suggestion that there were already improvements and elevations of A beta 42/40 ratio, which is suggestive of a reduced plaque load in the brain. But again, more trials or bigger trials are needed for establishing any effects. And here they design a foldamer, which is a big molecule that actually can bind to proteins and help these proteins fold. And when they did that, they designed that and they found that they could improve or help to prevent Alzheimer in animal models. Here they show that amyloid beta actually disrupts the nuclear pore complex. So that's a big protein complex in the how to call it, membrane, right? That is sitting around our nucleus. So the nucleus where the DNA is, and these nuclear pores allow communication between the nucleus and the rest of the cell. So mRNA, for example, needs to come out of the nucleus into the cytoplasm to do its stuff. Proteins from in the cytoplasm need to go into the nucleus to, for example, bind to specific places in the DNA, in the genome and activate the genes. So those are called transcription factors. So there is this communication between the nucleus and the cytoplasm, and this is being disrupted by amyloid beta. And that then makes these cells more vulnerable to a type of cell death, which is called necroptosis. So yeah, here they look at the role of mTOR in Alzheimer's disease. So and here they develop a new biomarker for Alzheimer's disease based on the glycan modifications to proteins in the blood. So actually, this was first developed in Belgium for aging, for liver cancer, and now we have one for Alzheimer's based on N-glycomics. Here they scan the brain of people with MRIs, and they use those data to develop a brain age clock that is able to predict cognitive decline in Alzheimer's. Here they show that this DICOP1 protein, which is involved in Wnt signaling, is important for Alzheimer's disease, for amyloid beta Alzheimer's disease. And what it actually does is you see a decrease in canonical Wnt signaling and an increase in the alternative Wnt signaling. So there is a switch between the two pathways, from the canonical to the non-canonical Wnt signaling. And it's interesting because we will come to a later paper on Wnt signaling in regeneration. Parallel, so here what they do is they look at marmosets, a type of monkeys, which is an upcoming model for aging, and they wanted to know if these monkeys are showing similar changes in aging related to memory decline compared to macaques and humans. And indeed, they find that there are big similarities, so that the marmosets is a an animal that can be used for studying cognitive decline. And here they exposed rhesus macaques to radiation, and they found that lifespan was on average 5.1 years shorter in irradiated animals. And these animals also had an earlier onset of various diseases such as periodontitis, so inflammation of the gums in the mouth, cataracts, atrophy of the testicles, tumors, diabetes, brain lesions, and so on. And now we come back to the Wind beta-catenin that I discussed just before. And here they look at this species of Hydra, the Hydra oligarchis. And this one, in contrast to the Hydra that you are more familiar with, the one that is, immortal, this one actually has sexual reproduction, and when it has sexual reproduction, it actually ages. It can switch between that non-sexual and sexual reproduction and aging or non-aging. And the Wind beta-catenin canonical pathway is important in keeping the regenerative ability there. So just like before, we see that the canonical pathway is good, and then probably the non-canonical one is going to be bad. So here, this is also quite interesting. They looked at people's the cytokine profile in the blood. So cytokines are immune molecules that are inflammatory or can be inflammatory and anti-inflammatory, but here I think they only look at the inflammatory ones. So they find that these ones increase up to 4 years before cancer diagnosis in people over 80 years of age. So they suggest that there are these tumors already present, of course, before diagnosis, years before, and that these tumors secrete a high amount of these cytokines in the blood, and that contributes to age-related chronic inflammation, as seen here. Yeah, maybe this paper just shows that reactive oxygen species are important for plasticity of, so the regenerative capability of animals. Here they find that the proteome size is positively correlated with lifespan in mammals, but actually the opposite happens in birds where it's negatively correlated. So here, remember at a Euro Symposium, we had Hannah Holstege in 2016 or 2014, I forget, and she showed the data that in that Dutch supercentenarian woman, there were only, I think, 2 stem cell clones in the hematopoietic stem cells, so the blood-producing stem cells that were still that were responsible for like 60+% of all the cells in the blood. So almost all the blood cells came from just 2 stem cell clones. And the thing here that they show is that in the mice, this is actually not really the case. So while in humans, we see that decrease in the number of these clonal cells. So we see a clonal expansion, basically meaning that 1 or 2 stem cells are actually responsible for all of these blood cells, and in the mice you don't see that, so we don't see the clonal expansion happening. That's a difference between mice and human aging. And here they identify a specific type of hematopoietic stem cell that is important in aging, the CLU1, the CLU-positive, hematopoietic stem cells. Yeah, here what they did is they transferred the microbiome, so feces actually from one animal to another animal, and they did that from 8, so they took it from 8-week-old mice, so that was a young, or they took it from animals who were the same age as the animals to which this stool was given. So young transfer of old transfer of stool to animals. And what they found is that when you give young stool to animals, you show an improvement in coordination, and intestinal permeability was improved. And also inflammatory markers were done in the mice that were receiving young stool. So here they use ultrasound to actually rejuvenate senescent cells. So these cells lose their senescence markers, such as beta-galactosidase and P16 and P21 expression. Also, there are changes in the methylome, the DNA methylome here, and so on. So this suggests that these cells become more like normal cells again. And in fact, you could enhance it by treating with some compounds, including rapamycin. You know, I will be a little bit careful here because, of course, you know, these cells are senescent for a reason. So trying to unmake them from being senescent is a little bit of a risky move. But yeah, that's what they did. But what they also say is that when they treated mice, they actually had an increase in lifespan and health span. I haven't looked at the full text yet, so I don't know how much the lifespan was improved by this treatment. It's I think it's open access, so if you want, you can go and have a look at the paper and see if they have the lifespan curve there. Yeah, so here they use reprogramming for cells in the intervertebral disc, so in between the vertebrae. And when you get all of these discs degenerate, and so that leads to lower back problems and lower back pain. So they took these pluripotency genes, the OCT4, the KLF4, and the SOX2, the ones that the Yamanaka factors without the MYC, because the MYC is linked to cancer, so they didn't use that one here. So they took these three Yamanaka factors and they put them in exosomes, and they treated these cells, and they showed them that there was a reduction in senescence markers and so on. Here they looked at something like 8 or 9, whatever, different types of diets in the nurses' health studies and the health professionals follow-up study. So 2 big studies of where they just longitudinally track people and their lifestyles and health and behavior and their health. And so they did like 30 years of following up in these people. And what they found is that well, all the diets that they looked at are diets that are supposed to be health promoting diets, right? So the Mediterranean diet, the DASH diet, and things like that. And indeed, all these diets improved multiple markers of health, you know, healthy aging, cognitive function, the number of illnesses that the people had, and so on. So all these things were improved, but some diets improved them more than other diets. And it won't surprise you when you look at what the diets are that work very well. It's diets rich in fruit, vegetables, whole grains, unsaturated fats, nuts, legumes, low-fat dairy products, sorry. Those were all linked to better aging, and higher intakes of trans fat, sodium, and sugar beverages, red and processed meats were all bad. So nothing really new in that sense. Yeah, here they develop aging clocks for the human brain, but this was done, so you need to take brain samples. So this is not something you can do on living people. This is on dead people. Yeah, so in that sense, maybe not very interesting. But so here there are these migrasomes. Those are actually a type of vesicles. When cells migrate, they leave these vesicles behind in the extracellular matrix. And then when another cell comes by, it can take up that vesicle. And so young fibroblasts produce more of these vesicles than old fibroblasts. And when they took these vesicles and they treated older mice with these vesicles, they showed an improvement in skin wound healing. So that's interesting. So here there's a paper on the dog aging project. You can have a look at. There is no real conclusions in here, they just discuss all the data they have been collecting, such as, blood counts, chemistry profile, immunophenotyping, flow cytometry, and so on. So it's just what they have all collected. So yeah, here they show that the what is it? The Grimage, is it? Yes, Grimage 2 methylation clock is able to predict mortality in people having cardiovascular kidney metabolic syndrome. So let's see here. So they had 14 years of following up. There were over 1,000 people who died, and each 5-unit increase in this methylation clock of aging was associated with a 50% increase in all-cause mortality in these people, a 77% increase in cardiovascular mortality, a 42% increase in non-cardiovascular mortality. And when you compare the lowest Grimage with the highest, you saw a hazard ratio of almost 2 for all-cause mortality, so almost a doubling of all-cause mortality, a 3-fold increase in cardiovascular mortality, and a 1.65-fold increase in non-cardiovascular mortality. So Grimage is very predictive in this population of people. And here they look at the combination of statins with ezetimibe. And ezetimibe is a drug that prevents the absorption of cholesterol in the gut. And the combination of this was significantly better than either therapy alone in reducing cardiovascular disease risk. So here we had a significant reduced all-cause mortality with a hazard ratio of 0.81. There was also major adverse cardiovascular events, 0.82, and stroke incidence at 0.83. And it was insignificant for cardiovascular mortality with a hazard ratio of 0.86. So it's still a reduction, but it didn't reach significance. And here they show that ketones are important for the normal heart function during aging. Yeah, I'm going to skip that. Hemophilia is associated with accelerated biological aging, according to telomere shortening and the number of mitochondrial DNA copy number. And here, what they did is they induced a torpor-like state in mice. So it's a little bit like hibernation in mice. And they show that that slows epigenetic aging and improves the health of these animals. And then they try to isolate what was the cause of these improvements. And they found that it was actually the reduction in core body temperature that was driving this beneficial effect. We are going to skip that because those are C. elegans and reviews. You can have a look at that if you are interested. And then there are 3 other very interesting papers being published. 2 papers that I will here discuss side by side are 2 papers where they actually designed a system-level metabolic map of the worm. And this is so the first time that such a map has been created for a multicellular organism, so with multiple cells. It had previously been done in like bacteria, in yeast, and in human cells, but not as a full human. And so this is the first step to trying to develop such complete metabolic maps of higher organisms. So a further step will be to the mice and to the human. But we have work to do to get there. Oops. And then here, what they do is they engineered fat cells to make them use a lot more glucose and fatty acids. So they by upregulating UCP1, that's the uncoupling protein 1. So that's a protein that when you have a lot of that protein, the mitochondria are uncoupled, so they stop making energy and they make heat. So when you do that, you have fat cells that produce a lot of heat. So they consume a lot of energy and they produce a lot of heat. And they implanted these cells into tumors. And the idea was that these fat cells will basically starve the tumor cells because they are like taking up all the nutrients, and actually it worked. So the tumors got starved from energy by these hyperactive fat cells. And that was a scientific news for this month.